Jai Hind children, I welcome all of my students in today's live class. Children, today is our biology class. Remember that before summer vacation, we have completed our first chapter of biology, that is nutrition in plants. So today again, I'm going to revise the same chapter, and for revision, I'm going to share my screen. Children, in this chapter, we will discuss the following topics. In the starting, we will discuss an introduction of the chapter. Then, we will discuss about the nutrition. After that, the types of nutrition which are found in living organism. Then, in detail, we will discuss about the autotrophic nutrition in plants. And after it, the process of photosynthesis. And after photosynthesis, we will discuss about the conditions which are necessary for photosynthesis. And after it, the synthesis of food other than carbohydrates by plants. And then the heterotrophic nutrition in plants. Children, the heterotrophic nutrition in plants are found in following four different modes. The first one is the parasitic mode, which are found in parasitic plants. Then the insectivorous modes, which are found in insectivorous plants. And then the saprotrophic nutrition. And the fourth one is symbiosis. And at last, we will discuss about the replenishing nutrients in the soil. So let's start revising our chapter with a little introduction of the chapter. Children, as you already know that all living organisms require food to grow, to repair the damaged parts of their bodies and get energy to carry out life processes. You also know that the components of food that are necessary for our body are carbohydrates, proteins, fats, vitamins, and minerals. And these all together are called nutrients. Nutrition. The definition of nutrition is the process of taking in food by an organism and its utilization by the body is called nutrition. Children, here you have to remember that taking food in and after it utilizing that nutrients which are present in the food by the body to generate energy, that whole process is known as nutrition. And this process of production of the energy is not completed only by the single nutrition process. One more process are required to complete the production of energy in the body, and that process is respiration. So the energy from food is obtained by the two processes, that is nutrition and respiration. Now, the types of nutrition which are found in living organism. So mainly there are two types of nutrition. First one is autotrophic nutrition and the second one is heterotrophic nutrition. Here the definition of these nutritions are the mode of nutrition where the organism make food themselves from simple substances is called autotrophic nutrition, and such organisms are called therefore autotrophs. And the definition of heterotrophic nutrition is the mode of nutrition where the organism cannot prepare their own food and depend directly or indirectly on green plants for their nutrition is called heterotrophic nutrition, and such organisms are therefore called heterotrophs. Now, The autotrophic nutrition in plants. So, green plants are able to prepare their own food from simple raw materials, that is carbon dioxide from the air and water from the soil. Plants get energy for preparing food from sunlight. And this mode of nutrition, where green plants make food themselves from simple substances, is called autotrophic nutrition. Now, children, here you have to know that the meaning of the word autotrophic. The autotrophic word is made up of two words. The first one is auto, which means self, and the second word is trophism, which means feeding. So, the meaning of autotrophic is the organism who can prepare their own food. Now, the cells of green leaves and young stems of plants contain numerous green structures which are called chloroplasts. And the chloroplasts are green because of the presence of a green pigment which is called the chlorophyll. It is chlorophyll that traps energy from sunlight. The leaves take in carbon dioxide from the air through tiny pores which are called stomata. Children, here I have given the diagram of stomata. Just see the diagram. It have the different parts like the guard cells. Inside the guard cells, chloroplasts is there. The guard cells have two walls, the inner wall and outer wall, and the guard cells get surrounded by the epidermal cells. The guard cells control opening or closing of stomata. The water is absorbed from the soil by the root hairs present in roots. The water and the minerals absorbed by the plants are transported to leaves through xylem vessels that are like pipes, and these pipes run through the root, stem, branches and leaves. Here you will study in detail about the xylem vessels in your higher classes. Now, the next one is the process of photosynthesis. So, the meaning of the word photosynthesis is made up of two words. The first one is photo, which means light, and the second is synthesis, which means combination of components. So, you can say that the photosynthesis is a process in which carbon dioxide and water in the presence of light and chlorophyll are converted into food in the form of glucose and oxygen is released. So this process is known as photosynthesis, and here I have given the chemical reaction which occurs at the time of photosynthesis. So you can see that the carbon dioxide and water, they both are going to combine together, and in the presence of chlorophyll and sunlight it's going to convert into glucose, and along with glucose oxygen is also formed in this process. Now, using the energy from the sun, a chemical reaction takes place in the green parts of the plant in which carbon dioxide and water are converted into food in the form of glucose and oxygen is released. Since the synthesis of food occurs in the presence of sunlight, it is called photosynthesis. Thus, the photosynthesis combines the non-living components of the environment, that is water and carbon dioxide, in the presence of sunlight to form food. The food synthesized by the green leaves is transported to other parts of the plant by the stem. In most plants, this glucose is converted into starch and stored in leaves, stems, roots, etc. Now, the food that plants make is used not only by themselves but also by the entire living world. Besides, as you already know, photosynthesis helps to maintain the balance between oxygen and carbon dioxide levels in the atmosphere. Therefore, in the absence of photosynthesis, life would have been impossible. The simplest method of testing whether the photosynthesis has taken place is to find out if starch is present in a leaf. So students, I will explain you this activity at last, that how we can check that the photosynthesis process takes place or not. So we can go through a simple test, and the name of that test is iodine test. So I will discuss the activity at last. Now, the next, the conditions which are necessary for photosynthesis. So there are mainly four conditions which are necessary for photosynthesis to take place. The first one is presence of chlorophyll. Second one is presence of light. Third one is availability of carbon dioxide, and the fourth one is water. Again, children, I will explain you at last with the help of the activities how the chlorophyll, light and carbon dioxide, they are necessary for the occurrence of photosynthesis. So now come to the next, synthesis of food other than carbohydrates by the plants. The carbohydrates that plants synthesize during photosynthesis are made up of carbon, hydrogen, and oxygen, and these are used to synthesize other components of food, such as proteins and fats. Proteins contain nitrogen. Though air contains large amount of nitrogen, plants cannot absorb this nitrogen directly. They get nitrogen in two ways. The first way is soil contains certain bacteria, which is called rhizobium bacteria, that can convert atmospheric nitrogen into water soluble compounds. Plant absorbs these compounds along with water to get nitrogen. And the second way is farmers add fertilizers rich in nitrogen to the soil, and these are absorbed by the plants to fulfill the requirement of nitrogen. Now, we have the second mode of nutrition, that is heterotrophic nutrition in plants. So, the animals and the non-green plants such as fungi and bacteria, they cannot prepare their own food. They depend directly or indirectly on green plants for their nutrition. And this mode of nutrition is called heterotrophic nutrition. Animals and non-green plants are known as the heterotrophs. Children, as you know that the herbivorous animals depend directly on plants for their nutrition. Carnivores depends on other animals, which in turn depends on plants. Thus, when you eat meat, you are indirectly depending on food prepared by plants. You know that the green plants get energy to prepare food from the sun. Thus, the sun is the ultimate source of energy for all living organisms. Now, some non-green plants which live in or on other living organisms are derived their food from them. For example, the dodder. It is a plant that sucks food from another plant using root-like structure, which is known as ostoria. And such plants are called parasites. So the first mode of heterotrophic nutrition in plant is the parasitic nutrition, which are found in parasitic plants. Some plants like mistletoe plant which grow on trees such as mango tree or mahua tree have green leaves and can synthesize their food. They take water and minerals from the host plant only, and that is why they are known as the partial parasites. So, children, see that the parasites can be categorized in two categories: total parasites and partial parasites. Total parasites, they derive all of its nutritional requirements from host plant. The example is fodder, rafflesia, etc. And the partial parasite, as already I have told, these parasitic plants derive some of its nutritional requirement from host plant. For example, mistletoe. Now, the second kind of heterotrophic nutrition is insectivorous plants. So, the plant kingdom also has plants that consume insects. The Venus flytrap and pitcher plant. They catch insects by ingenious methods and they are known as insectivorous plants. In the pitcher plant, a leaf becomes modified to form a pitcher-like structure with a lid. Inside the pitcher, there are hairs pointing downwards. When an insect enters in the pitcher plant, the lid closes to trap the insect and it gets entangled in the hair. The cells of the pitcher plant secrete digestive juices to digest the insect. The Venus flytrap and the pitcher plant. They both grow in soil that is not so rich. They use the nutrition obtained from insects to supplement the food they prepare by photosynthesis. Now, the third mode of heterotrophic nutrition in plant is saprotrophic nutrition. Some non-green plants which live on dead and decaying plants and animals and derive their food from them. For example, mushrooms and other fungi and the bacteria. They secrete digestive juices on the dead and decaying matter. This converts the solid matter into a liquid. They then absorb the nutrients from this liquid. This method of getting nutrients from dead and decaying matter in the form of a liquid is known as saprotrophic nutrition. The fungi and bacteria that use this mode of nutrition are known as saprotrophs or saprophytes. Children, you also have to know that the spores of fungi are always floating around in the air and they start germinating when they land on wet and warm things. Fungi grow on shoes, cloths, pickles, and other articles in warm and humid weather. Now, the fourth mode is symbiosis. In this mode, the two different kind of organisms work together for their mutual benefit. For example, alga, which is an autotroph, and fungus, which is a saprophyte. They both live together in lichens. The fungus supplies water and minerals to the cells of the alga, while the alga supplies food to the fungus. And such a mutually beneficial relationship is called symbiosis. Now, children, at last we have the topic replenishing nutrients in the soil. So, the plants absorb nutrients from the soil. Therefore, the amount of nutrients in the soil goes on decreasing in a forest. These nutrients get naturally replenished by decaying of dead plants and animals. However, on a farm, these nutrients have to be added to the soil in the form of manure and fertilizers. These manures and fertilizers contain plant nutrients. such as nitrogen, potassium, and phosphorus. A farmer knows which nutrients are crucial for which crop and add manures and fertilizers accordingly. Most crops require a lot of nitrogen to make proteins. After the crop is harvested, the soil becomes deficient in nitrogen. You have already read the bacteria rhizobium can take atmospheric nitrogen and convert it into a soluble form that the plants can absorb. The rhizobium cannot make its own food. It therefore lives in the roots of leguminous plants like gram, peas, and moong, where it gets food and shelter. In turn, it provides them with nitrogen. It thus has a symbiotic relationship with leguminous plants. We get most of the pulses from leguminous plants, and the farmers know that they do not need to add nitrogenous fertilizers in the soil in which leguminous plants are grown. So, children, this is all about the revision part of the chapter. Now, as I told that about the activity of the chapter. So, we have an activity in the chapter that the starch formed in the process of photosynthesis. So, come to that. The objective is, our aim is, the aim of the activity is to test that starch formed in photosynthesis. So, to taste a leaf for starch. For this activity, we require some materials like we require a potted plant, we require the taste tube, beaker, Bunsen burner, iodine solution, alcohol, water, etc. So the procedure of this activity is, first of all, we'll take a potted plant and we will keep it in sunlight for about 2 hours. After that, we'll pluck a leaf from the plant and boil the leaf in water for about 5 minutes. So the setup will be, we will set our setup like that. We will put the leaf in water in beaker and will boil the water for about 5 minutes. It will kill the cells of the leaf. After that, we will transfer this leaf in a test tube and fill the alcohol in this test tube and place this alcohol in test tube with leaf in a beaker. Put water in the beaker and warm the water. We will heat this setup until the alcohol start boiling. Children, here you have to know that the alcohol will start boiling first, then water, because the boiling point of alcohol is less than water. So we have to only allow to boil the alcohol, not the water. So about 2 minutes, just take out the test tube and with the help of the faucet, take out the leaf from the test tube and spread it on the white surface. And after that, put some drops of iodine solution on it. And after putting the drops of iodine solution, you will observe that the leaf becomes blue black in color. So this indicates that by the process of photosynthesis, the starch formed or food formed in the leaf, and that starch gets changed its color blue black when the iodine solution kept on it. So this is the activity in which we taste that a leaf form the starch during the process of photosynthesis. Now, we have some more activities in the chapter. Remember that children, we have discussed that there are certain conditions which are necessary for photosynthesis. So, just come to them one by one. Now, the first I'm going to explain about the condition which are necessary for photosynthesis is light. So, the sunlight is necessary for photosynthesis and that we are going to show with the help of activity. So, the objective or the aim of the activity is to show that to show that light is necessary for photosynthesis. So, this is our objective to show that light is necessary for photosynthesis. Now again for this activity we require at least the same material that I have told in the previous activity. A potted plant, test tube, beakers, burner, iodine solution, alcohol and all. Here we will take the strip of black chart paper. So the procedure is select a leaf of the plant. As we have taken a potted plant here, so one of the leaf we will select and we'll take a strip of the black chart paper and wrap the half part, at least the half part of the leaf with that strip and apply the paper clips. So here this much part of the leaf I have covered with the black chart paper and applied the clip, and after that keep this whole setup in the sunlight about 2 hours. After 2 hours, plug this leaf and follow the procedure. First of all, we will boil the leaf in a beaker. In water. After that, after about 5 minutes, we'll transfer this leaf in a test tube containing alcohol. And and we'll keep this test tube in a beaker which have the water. And again we will warm it. When the alcohol start boiling about 2 minutes, we will keep out the leaf from the alcohol and place it, spread it on a surface. And we will put some of the iodine solution on it. So we will observe that the part of the leaf which was covered by the black paper strip, its colors not get changed, it remain the same color, and rest of the part get changed into blue black. So this indicates that the starch formed in uncovered part of the leaf, while in covered part of the leaf where light is not able to fall, the starch not formed, and as a result there is no change in color observed. So this activity shows that the light is one of the important condition which is necessary for photosynthesis. Now, after this, we have another condition which is necessary for photosynthesis, that is chlorophyll. So, come for that. The aim is to show that chlorophyll is necessary for photosynthesis. So, now, children, here in this activity, most of the steps are the same. Only the difference is that here we'll select a kind of leaf which have the spots. Like some part of the leaves are green, some of them the parts are other colors like red color, yellow color, white color, etc. Like the money plant leaf. The money plant leaf, you must have observed that it have some green parts and some yellowish part or white. So such kind of leaves are known as variegated leaves. Money plant, croton, etc. These are the plants who have such kind of variegated leaves. So first of all, we'll select a money plant leaf which have the spots of either the yellow color or white color. Rest of the procedure will be the same. First of all, we will boil it in water. About 5 minutes, and then we will transfer the leaf in a test tube which contains alcohol, and we will sift this test tube in a beaker of water and heat this water. When the alcohol start boiling, so after about 2 minutes, we will again take out the leaf and spread it on a white surface and put few drops of iodine solution on it. So we'll observe that the green parts of the leaf gets changed in blue black, while the rest of the part which are yellowish in color is remain same. So this indicates that chlorophyll is necessary for photosynthesis because this green part of the leaf have the chlorophyll, whereas the yellowish part does not have the chlorophyll, and as a result starch not formed in yellowish part of the leaf. Now, children, after this activity, one more activity we have in the chapter in which we can show that the another important condition which is necessary for photosynthesis, that is carbon dioxide. So our objective is to show that carbon dioxide is necessary for photosynthesis. Here in this process, we will take the same kind of materials and some more, like we will take here the potassium hydroxide and the same potted plant and a small glass jar with the lid of cork. So our setup will be like that. We will take a potted plant and select a leaf of the plant. We will take a small bottle with a wide mouth and put some of the potassium hydroxide solution in it and put the leaf inside the bottle. We will cover the mouth with the cork lid. Inside the bottle we have some potassium hydroxide solution. Its formula is KOH. Now keep this whole setup in the sunlight for about 2 hours, and after that pluck this leaf and one more leaf which was not covered and taste them for starch. So the process is the same. First of all, we will place both of the leaf in water and boil them about 5 minutes to lose the leaves. After that, we'll transfer both of the leaf in alcohol in test tube and sift that test tube in a beaker with water and heat that water in the beaker. And when alcohol start boiling about 2 minutes, keep out both the leaf and spread them on a white surface. Now put the iodine solution on both of them, and we will observe that the leaf which was kept inside the bottle not converted into blue black, whereas the leaf which was not covered in the bottle gets converted into blue black in color. So this shows that the carbon dioxide which is present in the atmosphere used up by this leaf and photosynthesis occurred, and as a result starch formed and get color change blue black in the presence of iodine. And in this leaf, when it was kept inside the bottle, carbon dioxide is not present here. Now, children, here you have to remember that why we have kept potassium hydroxide. So the reason is simply this jar contains some of the carbon dioxide. So to remove that carbon dioxide content inside the jar, here we have taken this chemical. The property of this chemical is to absorb the carbon dioxide. So whatever the carbon dioxide which are present inside the jar get absorbed by the potassium hydroxide solution. So there is no carbon dioxide at all inside the bottle. And in the absence of carbon dioxide, starch not formed in this leaf, and as a result its color not get changed blue black. So, children, this is all about the revision of the chapter along with all activities. I hope that you understood all the chapter properly. Again, we will now start our next chapter. So, children, be ready for the chapter next class. Thank you and have a nice day.